Swayam Prabha. Digital India. Educated India.
He is a prominent researcher and former professor of chemical and computational biology at the Center for BioTechnology at the Anna University in Chennai. His work merges experimental chemical biology and computational methodologies to push the boundaries of modern biotechnology. Some of his works include topics such as computational biology and bioinformatics, enzyme technology, and bio-inspired nanotechnology. Please join me in welcoming him.
Thank you. Thank you very much for your introduction. So, I haven't done any research or teaching for the last 7 years. I retired in 2019. I do not teach. I, of course, do some small experiments at home, which I will allude to and maybe the last slides as I go on. So the talk that I'm going to give you is a very personal one. I mean, it's future directions in biology, lessons from nature. It's my personal thing that I've chosen the examples that I give you because I think that it can be done in India. Okay, so this is the idea of this talk, and I hope I will inspire some of you to do this. One of the best quotes that I've heard in I've seen for science is this one by Max Planck. Experiments are the only means of knowledge at our disposal. The rest is poetry and imagination. So I will allude to it in my last slide, why he said this and how he did this and came up with his theory of E is equal to H nu. The other one for life is he who has a why to live can bear almost anyhow. Okay, so this is a very, very important quote from Friedrich Nietzsche in The Twilight of the Idols. And now the new adage in science is you must be the jack of all trades, but master of one. So before I go, these are some of the websites that you can really look at. I'm sure if this will be available. So one is openstacks.org. Have you heard of that? Oh, these are some of the best books in physics, chemistry, biology, arts, everything. And you should have a look at it. And the concomitant thing is libretext.org. So both of them nearly are complementary to each other. Very, very amazing book is Tree of Life by Michael Telford. It's available at amazon.in for about 432 rupees. And A Science of Biology. I'm very sorry. I just found out that it is freely available on the web. And you can actually download it. I'm very sorry that I couldn't get the thing. One great website you should all look, especially the girls here, or even boys should have a look, is www.lostwomenofscience.org. You can see how women have really done so much for science. Have you heard of something called Langmuir-Blodgett films? Have they taught you in chemistry or physics? Okay, these are monolayers. Basically, for example, the TV that you see, the LCD TV that you see, or the LCD watch that you wear is all an example of Langmuir-Blodgett films. And of course, Langmuir, you know, was a Nobel Prize winning chemist. He was the guy who gave the octet rule and gave the term octet. And Blodgett, I didn't know till about 3 months ago, was actually a woman called Catherine Blodgett. And she did all the work on this thing. So there are a lot of nice things. It's a 30-minute video. There are about 12 women scientists who are put in there. I would also like you to maybe subscribe to these two things. The scientificamerican.com, it's about 4,000 rupees on an e-subscription. National Geographic is about 3,000 on an e-subscription. Please do that. And you should also know a little bit of philosophy, I think. So, but please do read all this after your 12th boards. One is Philosophy of Natural Science by Carl Hempel. This is a freely available book, and you should especially read the second chapter. Okay, this is on the inductive method, and actually it gives you the example of a great medical doctor called Ignaz Semmelweis, because of whom maybe we are all living, because he gave the thing that what doctors should wash their hands before childbirth. Okay. Then the other one is Man's Search for Meaning by Viktor Frankl. Again, this sort of exemplifies Nietzsche's statement about why and how. So this is the structure of my talk: agriculture and sustainability, materials, computation, home garden, and healthcare. Usually I used to give healthcare the first one, but I think it's a little deeper. I gave it to college students. We'll see if we have time. I will see it, and if you are not bored, we will do it. So agriculture and sustainability. So this is something called enhanced rock weathering, ERW. Now that we are planning to go to the moon and Mars and mine, we will call it terrestrial enhanced rock weathering. So not many of us know that 66 to 67 million years ago, India had one of the biggest volcanic eruptions that the Earth has ever seen in its lifetime. Things that are 120 kilometers down were thrown up through a series of volcanic eruptions which lasted about 10,000 years. It was the largest volcano eruption, and you can see that the lava flow, it was centered around Mumbai. And the lava flow was 1,500 kilometers. It went all the way up to Rajamandri in the Krishna Godavari basin. And so these have a very typical, so KTB also says it's Krishna Tungabhadra basin. So you can see that they have a very nice structure, a very unique structure. They are in steps. So if you fly from Chennai to Mumbai, you will pass through Pune. The flight will take you through Pune. And if you look out of the plane's window, you will see this. This is near Pune. The picture is taken near Pune. And you can see that it looks like steps. You can see here that it is like steps. So these are known as Deccan traps, and it comes from the word trappsteg, from the Swedish word trappsteg, which means steps. So you can see that this is the kind of volcano eruption you saw. So I think, how many of you like chemistry? Okay. And I'm sure, thank you very much. And I'm sure in chemistry, as a subject, if you now classify it as physical, inorganic and organic, and now biological, I think the last one that you will ever like is inorganic chemistry, right? But no, you like inorganic? Oh, fantastic. And so we will do something on inorganic chemistry. So this basalt that is there in India is once covered an area of 3 million kilometers, square kilometers. This is much more than some of the big countries in the world. And it is now, due to erosion, it is about 500,000 kilometers, which itself is very good. Average thickness is 500 meters to 2 kilometers. And you can have a very nice review here in Princeton about the Deccan traps. You can actually go through it. So these are the ones, they have all kinds of things we will see in the next slide. But what we will see is that these are shown as ultramafic rocks or ultramafic. Yeah, so this is what this means is it's low in silica content, about less than 45%, high in 18% in iron and iron oxides. That's why the name mafic, magnesium and ferric. So this is called mafic. And you can also see that these ultramafic rocks also contain potassium oxide and sodium hydroxide. And sodium oxide. And so once they leach, you know, both K2O and Na2O are nice basic oxides. So you put them in water and you will get a basic solution. And this is very, very important. We'll say why it's very important as we go on. Yeah, if you have any questions, do stop me at any time. Okay. So now what we can do is that the way we do it is that we search for suitable resources and sites. Then we mine it. Then we grind it. Mostly we grind it to about 1 millimeters. Okay. And then we transport it and then we scatter it on the cropland. So this is how we can use. So this is actually known as basalt. So you will see that basalt is also used in construction. So if you go to a construction way, you will see that gray powder sometimes. Not the one, don't confuse it with cement and sand, but next to it there will be a gray powder, and that's actually basalt. So here is the typical kind of composition that you see in the Deccan traps. So you have silicon oxide, which is, as I said, less than 45%. TiO2, aluminum oxide, iron oxide, magnesium oxide, calcium oxide. So you can see that magnesium oxide and iron oxide, if you add it together, it's greater than 18%. So this is how. So this also gives rise to some great minerals that you can see. If you go there and maybe scout around for a long time, you can see that some of those minerals that you get are, for example, apophyllite, stilbite, and modernite, all these kinds of things. And then they also give very industrially important compounds like zeolites. Zeolites are used for cracking. They are used for oil recovery, for all these things. So they generally have a formula. This is the formula: aluminum oxide, silicon oxide with water. So these are some of the reactions of this. So again, the Indian basalt is very high in olive minerals and pyroxene minerals. So you can see that olivine contains iron silicate. Iron silicate, pyroxene, calcium, magnesium silicate, and they can take up carbon dioxide reaction with oxygen to give bicarbonate. We remember, we also said, what did we say? That it also contained potassium oxide and sodium oxide. So if there's water, what will you get? A basic solution. And in basic solution, what can you get? That you can stabilize carbonates and bicarbonates, right? So you can see that this is how it is done. And you can also see that some of these calcium magnesium get precipitated by calcium carbonate and magnesium carbonate. And you can also see that the half-life of calcium and magnesium carbonate is about 1 million to 100 million years. They slowly decompose. Okay, so all this are some of the reactions that you can see. Recently, what has happened is that if you go deeper down, you don't have oxygen. So it becomes an anaerobic process. Without oxygen, what you see is that this happens. Say, for example, iron silicate plus water gives you hydrogen. Magnesium silicate, iron silicate gives you a mixture and methane. So basically, this is the purest form of hydrogen that you can get, because there's no, usually hydrogen is made by cracking petroleum, but now you will see that it's actually made up of, this is totally an inorganic process of making hydrogen. So this is known as geohydrogen or white hydrogen, and now this is the whole thing, that if you drill quite deep down, maybe a kilometer or 2 kilometers, that you can actually get hydrogen in the basalt of India. So you can see here. So what is the other thing? If you had looked carefully in some of these reactions, you will see that a lot of this calcium and magnesium also go off as hydroxide and therefore become a little more soluble than the silicates. So what happens? These now become small. These become small metal ions, which are very, very important for growth of plants. And so you have now had about 3 different ways of uses of this basalt. One is actually carbon dioxide mitigation. The second is hydrogen formation if you dig deep down. And the third is actually release of small trace minerals, which are trace metals, metal ions, which are very important for the growth of plants. So the idea is that by 2050, India will be actually, if they use their basalt very carefully, they would be now sequestering about 1 gigaton of carbon dioxide. Okay, so this is a huge, huge amount, and so this is what happens. The next one is that this is a picture that which recently came in Scientific American, where they have used this basalt and then they first throw it inside distribute it inside the farmland. And once you do this, so this is near Bhopal, a village near Bhopal, and you can see what they have found out is that the crop yields have gone up by about 20% because of these trace elements getting into the system. There's another way. So we also saw that bicarbonate is what is the product. And now there are certain anaerobes in the soil which use bicarbonate. This is known as the reverse Krebs cycle. Have you heard of Krebs cycle? Yeah. Okay, what does the Krebs cycle do? Huh? No, the initial thing, what does it do? Glucose plus oxygen to give carbon dioxide and water, right? So if carbon dioxide and water were to give bicarbonate, so can you do the reverse one? That is, use carbonate here, come all the way by reverse Krebs, and then get succinyl CoA, which is succinic acid is CH2, COOH, right? So you actually got carbon compounds out of bicarbonate by reverse Krebs. So a lot of anaerobes do produce reverse Krebs. So this is very important. And for the very, very good, for the superb discussion on cycles itself, especially Krebs and reverse Krebs, please do look at Transformer, The Deep Chemistry of Life and Death by Nick Lane. It's available in Amazon for about 640 rupees. I mean, this is, I would say, Tree of Life and this particular book are very, very important. Whether you are interested in physics, chemistry, if you're interested in chemistry, this is a great book. If you're interested in biology also, this is a great book, but you should also read this. So, you're familiar with amino acids? Yeah? Okay. So, these are amino acids. Now, you're familiar with primary structure, secondary. Oh, then that's great. We can even, if you guys are not bored, we can do the last one too. So, my next example of materials is the spider web. The spider is not only a great engineer, but it's also a great geometria. Okay, so you have so many different kinds of spiders. Some of them are the funnel web spider, which is an exceedingly dangerous spider, lives in Australia and is the sting is exceedingly poisonous. There is something called the triangle weaver. What it does is it weaves a web like this, then has a very, very flexible, very flexible string of spider of the web. And then as the fly approaches it, it just releases itself. And this goes and hits the spider, hits the fly, and immediately this fly dies and it is able to do this. So there's a trap door. There's something called the augury face spider, where it builds a nice square or a rectangular web, and as the prey approaches, just puts it on top and eats it. There's also another spider called the diving bell spider. This is an amazing guy. He doesn't have a lung, doesn't have a gill, but still spends 99% of its time underwater. So how does this do it? So we'll come to it as we go on. So if you look carefully, how does the spider spin its web? So it has three amazing organs to do this. So you can see that here is where something called a spideroin is. This is the this is the silk gland, and the silk gland produces it, and then it is put into another bag, another organ, where it makes the silk proteins, and this is known as spideroin. Now it has to weave its web. So the web has to come as a string. So it has another organ, which is acidic, and then it pushes this spideroin mass into this so that it becomes a nice thin web, and then it uses its legs to take it out and spin it, and then it becomes like this. Why is it so important? Why is it now people are looking at it in a very, very different way? Because you can see, if you look at the spider silk, what you see is that it is made up of alpha helices, beta sheets, and also some crystalline regions. So this gives it so much strength. You would have heard, which do you think is the strongest material that you have seen, that you have heard of? Not seen, heard of? Huh? What? No. Where you can wear or something like that. Which is as a fiber, which is the strongest. Huh? No. As a jute, okay. Nylon, very good. Anything else? Kevlar, right? Kevlar is a bulletproof vest that you wear. And why are they very, very important? Because what you see is Kevlar. This is nylon 6, which is the nylon 6, and this is Kevlar. Kevlar is a polymer of 1,4-diaminobenzene and 1,4-dicarboxylic acid. So you just do a polymer, and then this is the polymer. The strength, and also the kind of, it should also be a little flexible, because when the bullet hits, through the flexible bonds, the energy should be dissipated, right? So you will see that this is dissipated through hydrogen bonds. Hydrogen bonds are a little more flexible than covalent bonds or ionic bonds. So you can see that this is how. So to make strong materials, you need hydrogen bonding. For it to, or materials which can dissipate energy, you need that. So this is how it's done. If you look at the spider fiber, you can see, or the spider silk, you can see how beautifully the hydrogen bonds are. There is an inter-hydrogen bonding, there's an intra-hydrogen bonding between each fiber, and therefore you can see that if you hit a bullet, if it hits a bullet or a bullet hits it, then you can it will be able to withstand that. That's why you will see that things that are much larger than the spider is able to sit on a spider web or get it. For example, a fly going at about 5 kilometers an hour hitting a spider web is just trapped. Any other material you use, it will just go through. So this is because of this kind of intense hydrogen bonding that is there in the spider web. But what is the big problem with the spider web? You can't get much of it, right? Yeah. So what can you do? So what the last few years, what people have found out is that due to convergent evolution, the organs that make the spider web, the organs in the spider, which makes the spider silk, there's nearly the same kind of organs that are present in the silkworm. Okay, so you can see here that if this is the spider web, it has a tail, the sac, this is the tail we said, then the sac where the spideroin is kept, and then the duct where it is long, and then that's one where the silk is pulled. Similarly, in the silkworm, you get a posterior silk gland, a middle silk gland, and an anterior silk gland. So this is exactly the way that the organs are there. Okay, so what can you do? Anything that we can do? Yes, sir. Oh, but nothing can beat this. I mean, the billions of years of evolution has actually given this. Of course, people are trying, but nothing to beat this, because see here, there's no chance of intermolecular hydrogen bonding. Sorry, intramolecular hydrogen bonding, right? There's only intermolecular hydrogen bond. But in the spider silk, you can see both remember we saw the in the previous this one. So you can see here that there is intramolecular hydrogen bonding. Between these two, there will be intermolecular hydrogen bonding, and therefore any kind of energy can be easily dissipated within this. Oh, very, very difficult. I mean, very difficult. It's possible, but you may be only able to make small stretches of it. Best is to use some nice other organism which makes it for you. Okay. So what can we do? We look at the spider machinery. What are all the genes that make up gives the spider silk, and see whether we can put it inside the silkworm. And that's exactly what people did. So this is what I said. Look at, this is known as the Joro spider. It's a crazy spider because it has now taken over half the world. I mean, it's an invasive species, so I think it's there in India too. And you can see it puts huge webs here. And you can see a bird can balance on that web. I mean, that's the strength of the of this web. And so this was done in China, and all the sorry, I forgot to tell you, all the references that I'm giving are all freely available. You can download it. Okay, I mean, nothing is paid, so you can just go to that. You can download this. It's all freely available. So that's exactly what they did. They took the spider, the genes that make up that are used for the synthesis of the spider silk, the DNA, put it in. There's a new technology called CRISPR-Cas. Then you put it in by microinjection. You can put the whole thing inside the silkworm egg. Here, and then you have to have a reporter. You have to show that only this particular, because there's a chance that this silkworm may not also make it may be a wild type silkworm and may not be able to, it's not a transgenic silkworm. And so you put a reporter called the orange protein. So whichever silkworm produces the spider silk actually glows red. So this is exactly this. It only glows red. The silk is still colorless. This is how it is spun. So now you can see that the whole thing can be spun. I mean, it's like a thing. I mean, so you can see this. So this is the strength of the spider silk. So there's a company called Craig Labs. You can just have a look at it. they have a new facility in Vietnam which makes 10 metric tons of this spider silk. You can see here that 6 grams of spider silk is holding a 80 kg tractor tire. So this is your strength that you have. And so people are also trying to now make these kind of things where they are looking at the strength of spiders. Can we now synthetically also do something and then with the genes and then produce stronger fibers? So this is, as somebody said, not new, as you said, not new synthesis, but can you now do something with the gene and then try to make a better silk? Okay, so that's possible. I'm sorry. So why is this so important to India? All of you have heard of Tipu Sultan, right? Okay, what did he do? One, leave the political aspect of it, the science aspect, the science and technology aspect. Huh? No, wood steel was known in Tamil Nadu, in Salem, much more earlier, about 2000 years older. This thing, I mean, it was known because the Salem steel had, that ore had molybdenum, so that actually gave strength to the steel. So once the molybdenum got completely gone, then you had the whole thing disappear. But he actually invented rocketry in India. And he actually he sent emissaries to China, and then got all these things. So when he went there, the emissaries also found out that area where they went, that that they were having a huge farms of silkworms. Huge farm. And then they also found out that the weather was very similar to Mysore. So they were able to smuggle some silkworm eggs, take it to Mysore, and then Mysore became a huge this thing of silk. And it was started by Tipu Sultan. And that's why you would be if you go around a sari shop, you will see Mysore silk, right? So Mysore is very, very well known for its silk. I mean, and there's a central silk board, silk institute. Research institute in Mysore. So we can sort of have this kind of a thing and actually do such things. So again, these silkworm. So now, as I said, now they have also started doing gene manipulation for the spider silk, make so many, so they can make recombinant silks, and then take it out, and then you can now manipulate it in vitro, outside the silk, and make it into different shapes. You can make biodegradable materials. You can use it for sensing materials, all this. So that was a very nice review. I think somehow it got, I didn't put it here. But if you want, please let me know. I will send it to you to my address. So this is the other story of the other great spider. This is called the diving bell spider. Doesn't have a gill, doesn't have a lung, but it has to have oxygen. So how does it do it? So this guy goes, stays underwater, makes a huge web between two plants, aquatic plants. The top of the web is very, very strong. So it's called the diving bell because it goes up once in a while, gets oxygen. I'm sorry, yeah, gets air. I wouldn't say oxygen, gets air, and then comes it and deposits it here. Okay, so you can see it deposits here. So people now looked at the structure. Why is it that the greatest enemy of air is what? Well, if you have air, it's inside something, and you want to displace this air, what would you do? Add water to it, right? Add water to it, the air will come. So this guy, so no water can ever stick to its legs when it takes this. So people looked at the structure of the, structure of the hair here, and you can see that it is very nicely spaced together, and it is exceedingly hydrophobic. So that no water can can get in there. So if no water gets in there, what can happen? The air will stay and you can take it up. So now I'll ask you a question. Can you use it for making a metal that floats on water? Huh? Yeah. Can you make a metal sheet that floats on water? Yeah. How? Anything? Yeah. The whole idea is you should not get water anywhere near that. Only air should stick. There is nitrogen and oxygen and a little bit of argon, but no water. Hydrophobic coating, little this thing. But because hydrophobic coating wouldn't solve the problem because there's no air. The air gives the buoyancy. We have to have air because that's what will make it buoyant. Porous also, no, it won't be hydrophobic, so the water will get in and push the air out. So this is a very interesting thing. So what happened is, with the advent of these crazy lasers that we have with very, very small line widths, what people have done now is that this again is a very nice paper. So geometry enabled recoverable floating superhydrophobic metallic tubes. So what you do here is take a metallic tube, this was aluminum, and then make small lines with laser. Remember, we saw that was about maybe that spider hair between each other, or maybe about 0.2 micrometers, right? So you make very, very small things here and then see what happens. And this is exactly what happened. See, you can see here that the air sticks, goes inside those lines, but the water is not able to get in. So they have made a raft out of this. You can see that the raft actually holds about 25 grams of a weight, all this. So and you can actually drill holes into it. Okay. And then you can see that you push it under water and it again comes up and floats. So this has been done with aluminum. So people say now you can actually do it with any metal. If it's with aluminum, you can make it. So will the next warfare be about unsinkable ships? Okay, so this is the kind of thinking that we see. So this is more a kind of thing that I just put in this particular slide because the guys who did this won the Nobel Prize last year. Yeah. So this is about using AI for looking at protein folding. Remember, we got the primary structure. The whole idea is to get the tertiary and the quaternary structure because functions are defined by tertiary and quaternary structures. And therefore, we should know, can we use AI and machine learning to actually look at, from a primary sequence, to look at the structure itself. So you can see here, these are some of the things. What are all the predictions? It cannot replace experimental structural biology because you have to know the structure to design a drug. And you can see that but it can help. For example, the best way now to look at structures is using cryo-EM. I don't know did you see the cryo-EM facility here? No? Oh, there's a cryo-EM facility in IIT. So you can see that this is it. And the predictions are perfect and always correct. That is not true. Will it predict interact with any drug? Maybe. Now there's a latest review which says it can. So what DeepMind has done, these guys won, the Nobel Prize was won by Hassabis and Jumper from DeepMind, Google DeepMind. And now they have started a pharmaceutical company They're bought over the pharmaceutical company. So wherever they do this prediction, they actually synthesize the molecule. Why I really wanted to tell you was that both the 2026 and 2025 Nobel Prize winners in chemistry were students of this man, Carl Fried. So it's very important to have a very good mentor. And the one was Professor Bavendi, who won the Nobel Prize for quantum dots, and the other was Jumper from Google Mind for this. And you can see that Carl Fried was an excellent teacher. When I was a postdoc in Chicago, I actually attended 3 of his lectures. I had some doubts in statistical mechanics, and he was a fantastic teacher. I mean, so you need such dedicated people to actually look at this. So you can also see that some of the structures that are not amenable for crystallography or EM, it does give see, for example, now it has looked at, they have used it for looking at the mysterious protein called PFCY-RPA, and from that they can develop this thing for a malaria vaccine. And also the thing is that we go on hearing in the newspaper that there are plastic eating bacteria, but the reactions are so slow. I mean, absolutely slow that you can't use it. So now you can use AI and ML to actually do this. So they have now sort of modeled something and come up with an enzyme called FastPET-ACE, which can do this. So all of you have a smartphone, right? So how, if you order a pizza, how does that guy know your location, and how do you know that what is the nearest pizza place? Any idea? Yeah. So your smartphone is actually programmed, that is always in constant touch with 4 satellites. Okay. So you can see that the nice photo that I took from NASA, and you can use triangulation and another additional thing to find out where, because it's 3-dimensional, you can use the other fourth satellite to do this. But what is the problem with this? There are 2 problems. What's the first problem? Yeah, all that is okay. So to do that, what should one do? That all your satellites should be synchronized, right? They should be. You should take into account all the things, you know. So it should be perfectly synchronized. And how do you do the synchronization? They are actually done by atomic clocks. So, for example, cesium is the primary standard. Rubidium is the secondary standard. And you can see that now you can actually have a secondary standard. You can buy it now in Amazon.com. It's not available in India, but you can buy it abroad or in a company, the rubidium standard, because they also use it for industrial applications in the thing. So you can use it. But then you will see that electronic transitions have some uncertainty. You know, Heisenberg's uncertainty principles play a problem there. But nuclear transitions don't have a problem. And you can see here that so researchers are now attempting to build the first nuclear clock. So what does it do? You have thorium, glass, and then you use a 148 nanometer laser to actually push the nucleon to the next higher level. So this is another thing. What's the second problem with GPS, especially that you saw in the Iran war? That the GPS was easily hacked by Israel. They were able to get in and then easily hack it. So you need a completely different way of looking at positions, right? So one great thing that has come up is what is known as quantum sensing. So the Earth's magnetic field offers a very good window. You can see that no two places on Earth have the same magnetic signature. It is because of even if you live in the building, because of the structures, the iron, everything that is used, and also the ground has a different magnetic property. So you can see so you can see that natural geology and this thing makes it up. So now there are a lot of things that have come up. One is, so you have to have a molecule that is very sensitive to magnetic fields. So one molecule that they have used is what is known as a molecule which has been genetically modified from the jellyfish. The jellyfish has a nice protein called the green fluorescent protein, which fluoresces green. Now they have made all kinds of analogs. You can have a scarlet protein, a scarlet fluorescent protein, yellow fluorescent protein, a red fluorescent protein, everything. So they made a scarlet protein, and then they put it with a molecule called flavin mononucleotide phosphate. This now reacts, it forms what is known as a free radical pair. So it donates an electron here, this becomes plus, that becomes minus. And now once this has become, it becomes magnetically active. You can now go to triplet states. You can see triplet states. So now you can see a triplet state, and then you put a magnetic field, okay, and then you will see that the fluorescence decreases. So depending upon the kind of fluorescence that you have in a building, you can have it as a small thing and go to building upon building, and looking at the fluorescence intensity, can you now find out which place you are in, right? So you can work all that out. So this is a big thing that has now come up. So these are all known as quantum sensing. So this is one of my last slides. So one is science, sustainability, and you. So what you have to do is always measure, measure, measure. Okay, so this is what I do at home. Now that I'm retired, I have to have some hobby, otherwise my wife will kick me out. So what we do is that we have a home garden, and when we pressure cook food, everything, we take that water and feed it to the plants. Peels are taken out, macerated in a blender, and we have a small home milli vermiculture pot where we put it and then do it. We also, since we do not know how much of pesticides are there in your vegetables, we also steam cook our vegetables in a idli cooker. So here you can see the drumstick inside an idli cooker, just taking out the thing. Some of the pesticides are water soluble, and it seems as if the millipedes and the earthworms love it. They really love this thing. So what you need is a notebook, maybe a screw gauge, vernier calipers, a measuring device. You can also have a UV torch, and a marker and a pen and a weighing scale. So if you are interested in cooking, or if your mother cooks or your cook cooks, I would suggest that you weigh the amount of rice, weigh the amount of water, and see how it gets cooked, and whether you can optimize your cooking by weight. Weight is much better than volume, because we generally try to do it by volume, right? We put one glass of rice and 3 glasses of water, but a weight, just for the fun of it, just try it once. There are other things that have come up with this. See, for example, we actually have a peace lily plant. So you can see that there are 2 flowers. This is the first flower that came in, then this is the second flower. After some time, this flower became green, and this is still white. And then you put this red flashlight, you can see the seeds in the green stem actually start glowing red. So it seems as if these guys also have chlorophyll. So do the plants need chlorophyll in their seeds so that the seeds can grow better? One never knows. So here you don't see it. The other thing that we found out was that when we put all these peels, we took this sand and then put it in isopropanol and put this in, and you see that it glows red. This red is due to the porphyrin or chlorophyll or bacteriochlorophyll compounds. This is because of this one, this particular ring. It's very rigid and therefore fluorescent. So it looks as if the earthworms and the millipedes don't like chlorophyll, and they eat everything else. They eat the fiber, they eat everything, but they don't like chlorophyll. So this is something that has intrigued me. So we are now looking at whether one can use this to look at the health of the soil, or also conversely look at the health of the earthworm and the millipede. Next, this thing, again, continuing what we do, is that I'm quite an old man, so now I have to look after my health. So I cycle daily. I do about 8 kilometers, and I cycle around BISAG-N. And you know that, so this is my cycle, which I use with a nice speedometer. And then we know BISAG-N. This is the great, Schmidt Memorial. And this is the road that is adjacent to the Smith Memorial. Then there's another road where we have the temple and the bus stand. So that's about 0.6 kilometers from there. And another 1.2 kilometers, you come into Shastrinagar and all this place. And of course, this is a window air conditioner I have at home. So I go on telling my friends that this is the best way to learn statistics. So you take your cycle, go, every 15 seconds, it gives you the temperature. Okay, so you can have about 100 data points. Look at what is the mean, what is the mode, what is the standard deviation, etc., etc. But there's also another thing that you can do. For example, the last few days, I have been doing this experiment. So, for example, this was a reading taken on 7th May, where I cycled between 10:15 and 11. I can see, so I call this road, the one that we saw adjacent to the beach, as 0. The temperature there is 108. So I come back, I do 2 rounds. So I come back, again, it's about 108. So error is as minimum as possible. 0.5 kilometers away from there, it is 106. And another 0.7 kilometers away, it's another 104. So for every 1 or 2 kilometers, it goes on reducing by about 2 Fahrenheit. So this is very, very nice. I mean, you can do such kinds of experiments. So the temperatures that you see or you live in is actually much, much different from what the weather station gives you. Okay. So the second one is that we have a window AC. The last few days have been abnormally hot and humid. I mean, more humid than hot. So you can see here, put on the AC at 125, and the temperature is 31 degrees. Humidity is 77%. And then at 140, it is 29.1, but the humidity has come down by about 20%. So you can see here that, and after about another 20 minutes, you can see the humidity remains at 58, but the temperature goes on and down. So is it that the air conditioners in India, in Chennai, are so designed that you, because I keep it only at 25, because I don't go below 25. So is it designed that humidity is the most important thing? So you just get the humidity down, and then the temperature is. This is according to me, this is the ideal thing. If you reach this, you can live happily. I mean, you can do a lot of work happily with this temperature, about 27 degrees, and this thing. So another thing that we have seen is that we also have this Karpuravalli plant, So this is Pennathur ambionicus, and an osmium, which is tulsi. You can see these two plants have no fungal or bacterial attack. And you look very closely, I mean, you can see that there are tiny hairs on the leaves, tiny hairs on the stem. So is it that these guys, attack the fungus or the thing that land on them and make sure that they die? And you can also see that once you have this, for example, in each we have about 2 balconies, and in each balcony we have 4 plants of this and about 1 plant of that interspread. You can see that we don't have any mosquitoes at home. So the one thing that we want to see is that does red light increase the production of volatile products? So we sort of constructed something like this. A student of mine who's an associate professor at Nagpur is now planning to do this. Take this, can be collected and analysis done. We can use gas chromatography and mass spectrometry to find out whether these volatiles are actually mosquito repellent or not. And can the fine hairs or stems and leaves be collected and microscopic studies carried out on them to find out the microstructure and look at how we can use it for human this thing. So the other thing is now the health of a plant is known by the amount of chlorophyll it has. So, for example, there's a nice company now called Petiole Pro, and what they have is a handheld smartphone from which they are able to look at the amount of chlorophyll and do it. I mean, it's a paid subscription. They have a color scheme from which you have to read out and say that this is the amount of chlorophyll you have. So you can see that you did Beer's law, right? Did you do something called Beer's law? To find out the amount of compounds that are there. Okay. So there's something called Beer's law. This is called absorbance, where you can, from the absorbance, you can find out the concentration. So the equation is that is, if this is I naught, P naught, we call it, and this is the incident ray, this is the transmitted ray. It is related to the concentration as in a logarithmic fashion. That is log P naught by P is equal to epsilon CL. So now, but if you use fluorescence, you can see the fluorescence intensity is proportional to concentration. So it's not a logarithmic effect, but this is a. So can you now have that red light, shine it on this, and then develop an app by which you can quantitate the fluorescence? So these are some of the things that one of my students is doing. Can I take another 10 minutes, or are you very tired? You can be very interesting about it. Huh? Huh? Yeah? I can or no? I can? Okay. Another 10 minutes, okay? I'll do. So the last one is healthcare. How many of you are interested in medicine? Well, put your hands up. It's okay. Medicine, biology, biomedical, all this. Okay, great. So what do you think is the most important drug that has come out in the recent years? No, that is a local thing. No? For more general, more. Oh, that's all done now. Huh? No. The latest drug that is now revolutionizing health. No, those are very specific. I'm saying a non-disease state, a lifestyle state. Fantastic. Ozempic. That's now taken the world by storm. You know what's Ozempic? Ozempic is a drug that reduces weight. Okay. And also And not only weight, what people have seen is that it also reduces cardiovascular disease, it reduces neural diseases, it reduces kidney diseases, and they don't know why. Still, they don't know why. You know how Ozempic was discovered? This guy was the saved us. This guy. You know what that is? Gila monster. Fantastic. So the Gila monster saved us. This is the beginning of Ozempic. Okay. The Gila monster lives in Arizona desert, and their food is very, very scarce. So whenever it eats, it has to eat, store it, and make sure that it does not digest the food very fast, right? It has to have a slow release that of glucose. Otherwise, if the stores are all gone, it'll be dead. So all of us have that, right? So all this you know. So we'll go off. Here, I just want to tell you two things. Have you heard of the single letter code for amino acids? Right? You know who did that? Who found out, who sort of said, let me use single letter codes? Correct. Margaret, day off. Because she was both a computer scientist as well as a biologist. So she was inputting all these protein sequences, and she said, I can't put everything together. So she got a whole range of, she made this. For example, proline is known as P, blah, blah, blah. And the man who did the sequencing of this, of the proteins, was a man called Sanger. He won the Nobel Prize for both sequencing the protein as well as the DNA. So how do we store glucose? Where do we store and how do we store it? Where? As glycogen in the liver, right? So glycogen is a polymer of glucose, right? So this is the structure of glycogen. And so when you hydrolyze it, you have stored it. So now you have to have a hormone which tells you when to release it, or when the enzyme should act on it to hydrolyze it, right? So that is done by this called a glucogen. A glucagon. Okay, so that's a 29 amino acid polypeptide. But the half-life of human glucagon is very, very small. So for people who are obese, the problem is that after some time, that's it. Glucagon is not released, and therefore glucose goes on getting stored, and then they have obesity. So how do we do this? How can we stop this? We can have a glucagon or a glucagon-like peptide, right? Which does not get degraded very fast, right? And there's a slow release of insulin, so that all the sugar that is there gets metabolized. So this is how we should look at the problem itself, and that's exactly what our man gave us. So a glucagon-like peptide is released from the gut. So you can see that so also inhibits gastric emptying and food intake, actions maximizing nutrient absorption while limiting weight gain. So again, I think once you have this thing, please look at all these references that I have given are all free. You can just download it. I mean, you may get some great ideas. I think youngsters give us more great ideas than us. So this is the GLP story. Oh, shoot. So you can see here that the Gila monster gives us this is our human GLP, okay, the glucagon. Okay, so this is extendin, which has a which is from the Gila monster. So you can see that both of them nearly have the same structure. So the half-life of very similar compound. For the human, it is 2 minutes. So the glucagon from the human, GLP from the human is given, and within 2 minutes it degrades, and that's it. The one from the Gila monster, very similar, is 13 hours. So what they did was that, and they also found out that most probably it was because enzymes cut this particular amino acids, the alanine and the aspartic and the glutamic acid. So what they did was that instead of having alanine here, they put what is known as aminobutyric acid. It's nothing. You know the structure of alanine? It is CH3H, NH3 plus, COO minus. So you take out that CH3, you have that CH3, this H you take it off and put CH3. So how many carbon atoms are there totally? 4. So it is called isobutyric acid. So it is called amino isobutyric acid, or AIB. So then you put a small, so you have to also make it a little more flexible. So you put a little linker so that can go into the enzyme, and then that's what they did. Just if you had followed day before yesterday's paper, you would have seen a newspaper. You would have seen that Lilly has come up with a drug candidate called retatrutide. Okay, this is supposed to reduce obesity by 28% in 3 months. The problem with Ozempic and all these kinds of compounds is you also lose muscle mass. But with retatrutide, you will see that they not only you don't lose muscle mass, you can have an injection which makes sure that you don't lose muscle mass. So this is supposed to be the 5th generation drug for obesity, cardiovascular disease, neural disease, and kidney disease. I told you about, just maybe the last slide. Okay, I told you about this Planck's great statement, right? And you must have all heard of Kirchhoff, right? Kirchhoff laws of electricity, correct? But Kirchhoff was a great physicist. Not only was he into electric thing, but also into spectroscopy, especially black body radiation. And he looked at black body radiation, looked at temperatures, exact temperatures, found out what kind of electromagnetic radiation was emitted by the black body. He wasn't very satisfied with the spectroscopes that were there, and therefore invented his own spectroscope, did all the measurements beautifully, and then came up with this. Boltzmann introduced the concept of states, right? He gave the statistical interpretation of entropy, and then he gave this. So what Planck had was Kirchhoff's great experiment on black body radiation. He knew about Boltzmann's states, Boltzmann, though people thought that he was a theoretical physicist, was a great experimental physicist too. So he had this. So now, once he got the black body radiation thing, looked at both the experimental, theoretical. He said continuous energy is a problem. It's not continuous. It's discrete. It has to be discrete. So that was poetry and imagination. He had these two experimental results, but what he did was poetry and imagination. So he came up with this and actually calculated what was the value of H. And he came up with this value, 6.55 into 10 power minus 13 joules second. It's of course not energy, it's actually action. And it's within 1.2% of the currently experimental value, but he did it by hand. And why is it H? I was always wondering, why is it H? It should be like P or something. It's the Planck's constant. He said H is Hilfsgröße. In his first paper, he mentioned it's Hilfsgröße or auxiliary energy. This is something that I want all of you to do. So this is some 3 questions I thought each one of you can answer. If you can take it down, you just go around any shop and you will see that when you go to a fruit shop, you will always see that, notice that bananas are the only fruit that are hung like this, right? And basically they are also hung in front of the shop, never at the back, the front of the shop. Why is it so? Huh? So what?
So where they're kept there, where they're kept on a basket or something, it gets accumulated at the bottom and that part rises faster.
Ethylene is the right answer, but why like this?
When they hung, it goes to all the parts of the banana, so that it rises even.
No. You have to take off the ethylene. You can't have the ethylene. Right? If you have ethylene, then it will become dark, so you have to take off the ethylene. Yeah? Correct. So you have to take off the ethylene. So how would you take it out? There's a problem with ethylene. What's the problem with ethylene?
It's a gaseous compound.
Yeah. Ethylene has the same molecular weight as nitrogen, right? 28. So that's why it's just a little lighter than air. Just a little lighter. Okay. And therefore the air, so you have hang it in the front, have fans there so that it takes off the ethylene. So can you use it for something else? Think about it and answer. Okay. The second one is, when you go one nice night, look at the sky, maybe a new moon night. Why don't you never see green stars? Green stars. You see green planets and green celestial objects, but never green stars. Why? Okay, this is the second question you have to answer. Huh? Yeah? What? No. It's a sort of a very confusing, it's not a straightforward answer. Okay, that's why I want you see red, blue, orange, white, yellow, all kinds of stars, except green. You see green planets. Uranus is green. And that's because of methane. And you see the new comet that came about 2 years ago, which was green, and that's because of CH2. So planetary objects are okay, but why stars are not green? Just have a look. And can you estimate pi by a toss of coin? Okay, you toss a coin. Okay. Huh? No. How to calculate pi from there? Huh? No, it's a very nice answer. So please try. I mean, you can use ChatGPT. You will never get pi from there. You may get, but this is a new one that you can try. But you have to have a large number of people to do this. Okay, you may get about 2.8 or something like that. But actually, you have to do it 3 billion times, 3 trillion times to get 3.11.14. Okay, but at least in the class of 100 people, you can toss and do this experiment. So just go through all this. Okay, Write it. Don't send me a typed answer. Write it, scan it, and send me by email to my travelpennathur@gmail.com. So, acknowledgements. I have to thank my wife very much, because she puts up with all my idiosyncrasies. All the things that I do at home, she's able to sort of say. Sometimes, of course, she gets very mad and says, "You please go to work again." But the university will not take me, so I better be at home. Dr. Ketika Garg, who is my daughter-in-law, she goes through all my slides patiently, gives me ideas and things, and do that. And my son Anuj Pennathur, who usually is very, very critical about all the things that I say, as most sons are, I guess. Thank you.
Thank you, sir, for your very interesting talk. The wide range of topics, from the minerals present in the Deccan Trap to different types of spider webs, protein folding using AI, and healthcare, kept us really engaged throughout the session and taught us many new things. The hands-on experiments were also very interesting to learn about, and we'll definitely ponder on the questions you've given. Thank you for your time and effort, sir.
Thank you.